Hello everyone. Today we're going to talk about animal phylogeny. Phylogenies developed from different molecules sometimes suggest quite different evolutionary relationships among the same groups of animals. However, combining data from multiple genes has resolved the relationships of most phyla. Although they differ from one another in some respects, phylogenies utilizing molecular data shared some deep structure with the traditional animal tree of life. Figure 1 is a summary of animal phylogeny developed from morphological, molecular, life history, and other types of relevant data. Some parts of this phylogeny are not firmly established, and new studies are constantly appearing, often with somewhat different conclusions. It is an exciting time to be a systematist, but shifts in understanding of relationships among groups of animals can be frustrating to some. Like any scientific idea, a phylogeny is a hypothesis, open to challenge and to being revised considering additional data. They symbolize autapomorphies with blue rectangles in the lineages of the unities they characterize. Molecular data support what had been the majority view that phylum Porifera is monophyletic. Among remaining animals termed Eumetazoa, molecular data are in accord with the traditional view that cnidarians branch off the tree before the origin of animals with bilateral symmetry, the bilateria. Our understanding of the phylogeny of the deuterostome branch-off bilateria has not changed much, but our understanding of the phylogeny of protostomes has been altered by molecular data. Annelids and arthropods, which had been considered closely related because both exhibit segmentation, are in fact not close relatives, but rather belong to separate clades. Now, arthropods are grouped with protostomes that mold their cuticles. These are termed ecdysozoans. Molecular sequence data can help test our ideas of which morphological features reveal evolutionary relationships best. In this case, molecular data allowed us to see that, contrary to our hypothesis, segmentation seems to have evolved convergently, but molting did not. But not all features are easy to diagnose, and molecular data do not resolve all uncertainties. The enigmatic phylum Ctenophora has been considered both diploblastic and triploblastic, and has been thought to have both a complete gut and a blind gut. Classes, orders, and phyla do not reflect reality. One must free oneself from the restraints of traditional classifications. Systematics is offered as a scientific product to those who would maintain open-mindedness in such a form that arguments for every single decision are comprehensible and checkable and can therefore be fully analyzed. The decisions made in phylogenetic systematics are not subjective. Phylogenetic systematics has a clear methodology which allows all its statements to be presented in the form of objective hypothesis. Any hypothesis can be checked at any time and by anyone, and thus can be either confirmed or rejected. Current understanding of animal phylogeny requires a rethinking of how some characters have evolved. Zoologists previously inferred that the first animals are acoelomate, that some of their descendants evolved a pseudocoelom, and that some pseudocoelomate descendants evolved the coelom. This view was so widespread that classification systems were based on the state of the coelom. However, evolution rarely occurs in such a linear and directional way. Our understanding of animal phylogeny now makes clear that the state of the coelom has evolved many more times than previously realized, and consequently that it is not a reliable character to infer phylogenetic relationships. A coelom appears to have evolved just once, in the ancestor of the clade comprising protostomes and deuterostomes. Subsequently, the pseudocoelomate condition arose several times from coelomate ancestors within the protostome clade. As a result, all deuterostomes have coeloms, but the condition in protostomes is mixed. In addition, although the acoelomate condition is the ancestral state for animals, some animals have lost the body space, becoming acoelomate secondarily. Two clades of protostomes are recognized as having evolved independently since ancient times, the spiralians and the ecdysozoans. Spiralian animals grow by gradual addition of mass to the body and undergo spiral cleavage. Lophotrochozoans move by muscular contractions and include most protostomes with a coelom. Most platyzoans are acoelomates and move by ciliary action. Ecdysozoans are animals that molt, a phenomenon that seems to have evolved only once in the animal kingdom. Of the numerous phyla of protostomes assigned to the Ecdysozoa, Arthropoda contains the largest number of described species of any phylum. Deuterostomes consist of fewer phyla and species than protostomes. Echinoderms and chordates share a mode of development that is evidence of their evolution from a common ancestor. For most of the 20th century, the accepted relationship between the three deuterostome phyla placed the hemichordates as sister group to the chordates, to the exclusion of the echinoderms. This was originally based upon a putative homology between the notochord of chordates and the stomochord of hemichordates. This putative homology gains no support from gene expression analysis. One possible relationship between the three deuterostome phyla groups, echinoderms with hemichordates, this clade is referred to as the Ambulacraria. These two phyla share a similar larval morphology, although Netzlin argued that these similarities may have originated by convergent evolution. Phylogenetic analysis of mitochondrial and 18S DNA sequence data and a combination of the two offered reasonable support for this grouping. Of particular importance are the chitin cuticle and ecdysis during growth. Raven et al. List paired, jointed appendages as a key characteristic of Arthropoda. However, the division of the appendages into articulated limbs, a second state of new features that evolved later, does not contribute to the characterization of the Arthropoda, but are rather autapomorphies of the subtaxon Euarthropoda. The cuticle, with its antiparallel arrangement of neighboring chitin chains and their covalent connection to protein, is secreted by the ectoderm. Once produced, the cuticle cannot be enlarged. Therefore, to grow, an individual must molt at intervals and produce a new cuticle. The loss of mobile cilia on the surface of the body is a consequence of the evolution of a hard chitin cuticle. The head of the arthropoda is formed from the acron and probably the first 3 segments with appendages. A dorsal pericardial septum spans the body horizontally. Hemolymph circulates in arteries that exit from the heart and sinuses of the body cavity. Veins are absent. Mollusca. Coelomate body of many mollusks is covered by one or more shells secreted by a part of the body termed the mantle. Many kinds possess a unique rasping tongue, irregular. Members occupy marine, terrestrial, and freshwater habitats. 35,000 species are terrestrial. Chordata. Each coelomate individual possesses a notochord, a dorsal nerve cord, pharyngeal slits, and a postanal tail at some stage of life. In vertebrates, the notochord is replaced during development by the spinal column. Members occupy marine, terrestrial, and freshwater habitats. 20,000 species are terrestrial. Flatworms, unsegmented, acoelomate, bilaterally symmetrical worms. Digestive cavity has only one opening. Tapeworms lack a gut. Many species are parasites of medical and veterinary importance. Members occupy marine, terrestrial, and freshwater habitats, as well as the bodies of other animals. Nematoda, pseudocoelomate, unsegmented, bilaterally symmetrical worms. Tubular digestive tract has mouth and anus. Members occupy marine, terrestrial, and freshwater habitats. Some are important parasites of plants and animals, including humans. Annelida, segmented, bilaterally symmetrical coelomate worms with a complete digestive tract. Most have chaetae on each segment that anchor them in tubes or aid in crawling. Occupy marine, terrestrial, and freshwater habitats. Cnidaria, radially symmetrical. A coelomate body has tissues but no organs. Mouth opens into a simple digestive sac and is surrounded by tentacles armed with nematocysts. In some groups, individuals are joined into colonies. Some can secrete a hard exoskeleton. The very few non-marine species live in freshwater. Echinodermata, adult body, pantaradial in symmetry. Water vascular system is a coelomic space, and the skeleton of calcium carbonate plates. Many can regenerate lost body parts. Fossils are more diverse in body plan than extant species, exclusively marine. Porifera, bodies of most asymmetrical. Defining an individual is difficult. Body lacks tissues or organs, being a meshwork of cells surrounding channels that open to the outside through pores, and that expand into internal cavities lined with food-filtering flagellated cells. Most species are marine. 150 species live in freshwater. Bryozoa, the only exclusively colonial phylum. Each colony comprises numerous small, coelomate individuals connected by an exoskeleton. A ring of ciliated tentacles surrounds the mouth of each zooid. The anus lies beyond the lophophore. Rotifera, small pseudocoelomates with a complete digestive tract including a set of complex jaws. Cilia at the anterior end beat so they resemble a revolving wheel. Some are particularly important in marine and freshwater habitats as food for predators such as fishes. Nemertea, protostome worms notable for their fragility. When disturbed, they fragment in pieces. Long, extensible proboscis occupies their xylem, that of some tipped by a spear-like stylet. Most marine, but some live in freshwater and a few are terrestrial. Tardigrada, microscopic protostomes with 5 body segments and 4 pairs of clawed legs. An individual lives a week or less, but can enter a state of suspended animation in which it can survive for many decades. Occupy marine, freshwater, and terrestrial habitats. Brachiopoda, protostomous animals encased in 2 shells that are oriented with respect to the body differently than in bivalve mollusks. A ring of ciliated tentacles surrounds the mouth. More than 30,000 fossil species are known. The stem species of the arthropoda was an aquatic organism. The transition of the Onychophora to terrestrial living must be interpreted as a case of convergence to the evolution of terrestrial Euarthropoda. In connection with the change to land came the development of tracheae. However, this cannot be homologized with the tracheae of Euarthropoda. Sickle-shaped blades open in the oral cavity. Stubby appendages without claws. A large, lensed eye lies dorsally on both of the antennae bases. Pairs of salivary glands open into the oral cavity. Ctenophora, gelatinous, almost transparent, often bioluminescent marine animals. 8 bands of cilia, largest animals that use cilia for locomotion. Complete digestive tract with anal pore. Ketonatha, small, bilaterally symmetrical, transparent marine worms with a fin along each side. Powerful, bristly jaws and lateral nerve cords. Some inject toxin into prey, and some have large eyes. It is uncertain if they are coelomates, and if so, whether protostomes or deuterostomes. Loricifera, tiny marine pseudocoelomates that live in spaces between grains of sand. The mouth is borne on the tip of a flexible tube discovered in 1983. Cyclophora, microscopic animals that live on mouthparts of claw lobsters, discovered in 1995. Micrognathozoa, microscopic animals with complicated jaws, discovered in 2000 in Greenland. Scientists have defined phyla based on tissues, symmetry, characteristics such as presence or absence of a coelom or pseudocoelom, protostome versus deuterostome development, growth pattern and larval stages, and molecular data. Among protostomes, spiralian organisms have a growth pattern in which their body size simply increases. Ecdysozoans must mold to grow larger. The condition of the coelom and the occurrence of segmentation have evolved convergently among animals, whereas other traits, such as molting, have arisen only once. Among the deuterostome phyla are echinoderms and chordates. Thanks for listening. See you next time.